Perfect. Great. So I'll go even faster through the epidemiology.
Just to check, can everyone see and hear us okay, and also see the slides?
Yeah.
Perfect, thanks. Sorry. Yeah, so this is it. What I'm going to talk about is about why I was looking at age of infection in particular as a ubiquitous exposure, which might be the kind that you can't pick up with conventional epidemiological studies. I'm going to then say why this is important. I'm then going to talk about age of infection itself and then the opportunities in the UK. So a basis for thinking there might be some very important etiological dark matter, if you like, hiding from sight. One goes back to thinking about heritability, people who are involved in genetic studies, especially in genome-wide association studies. This was a famous piece in Nature in 2008, when the first lot of genome-wide association studies were done, and they found very little indeed. And it was then there was this great mystery: how can it be that these diseases are heritable, highly heritable, but we're not actually picking up any of the heritability? And it turned out that it was basically because the sample sizes were too small, because people weren't able to include structural and rare variants, etc. And there isn't any missing heritability, so it's not an issue. But there is missing environment. Epidemiology has been incredibly poor in actually identifying environmental contributions to disease that we know must exist. Now, in the case of genetics, we have a gold standard that we know show that the genetics are important, which are twin studies. They tell us that we do know that this phenotype is heritable to this extent, so we should be able to find the germline genetic variants that predict it, which we now can. Now, there isn't the equivalent of heritability for environment, but for environment, we do have some very good ways of knowing what, in principle, must be preventable and must be environmentally caused. Just to say that when I say environment refers to all modifiable non-germline genetic causes of disease. So environment has been used very broadly. It isn't just sort of lead in the water or air pollution, but includes diet, smoking, everything, basically everything that's external and can be modified. And in epidemiology, the codification of this notion about saying to what extent is disease environmental and can it be modified goes back to Richard Doll and Richard Peter in 1981 when they produced the report for the United States government on the causes of cancer. And they put together data on time trends. So if something is changing over time, either increasing rapidly or decreasing, then that cannot be due to germline genetic changes. There must be some externality that must be generating those changes that must, in principle, or be intervenable. Even more powerful in a way are migrant studies, when people move from one population to another, and then you identify when they start having the risk of the population they move to. Is that seen in the single generation? Or is it seen only in second generations, etc.? But again, they tell you that there must be some modifiable factor, something which is changing, which influences disease risk. Geographical differences are clearly not explicable to any sensible degree by genetics. And you also have identified causes. Smoking was about the only, and asbestos were the only two actually identifiable causes. But they put these data together to say what proportion of each cancer type must be preventable in principle, i.e. if all people had the risk of a lower risk population, what proportion of cancers would be prevented? And they came up with this now sort of widely replicated and believed notion that 80% of the cases of cancer in both sexes are avoidable in principle, but only for a third was their cause. No, that's the distinction there. It's preventable in principle. Time trends, migrant studies, geographical differences, etc., tell us that there must be something that could be modified and change the risk, and what we actually have a known cause for. And they speculated on the causes of the avoidable in principle cancers. And for what was...
George, your slides aren't moving.
Oh dear.
We're stuck on the title slide.
Sorry. I'll carry on talking while the slides become animated. So they speculated on what the causes were, and for what were reasonable hypotheses for the time, with cancer as the outcome, diet was seen as a factor that fitted much of the bill. Infection was put down as being a sort of as something that they really couldn't know. Are they moving now?
Yes,
So,
yes,
yeah.
thank you.
So this was purely speculation, but on what they thought they would see, and the sort of success of epidemiology with smoking, with asbestos, to an extent with alcohol, etc. At that time, said, Richard Doll said in 1977, "I see no reason why causes for the remaining common cancers should not be detected within 1 or 2 decades, because there was clearly external factors that were influencing those cancers, and the epidemiological methods were working or seem to be working. So let's just go out, do lots of massive prospective studies, measure everything, follow people up, and we'll get the causes of cancers." Now, there's nothing special about cancers. It's just that cancers, you have cancer registries, so you can do these calculations. With global burden of disease, I'm working with some people in GBD to try and to expand this sort of notion to other diseases. It is not just that cancers are preventable, it is most common diseases, or most diseases are preventable in principle. But we haven't obviously found many new causes of cancer in the last 40 years. This is it's a very good paper by Richard Piso, Richard Doll, others in Nature in 1981, which might beta-carotene be something that would reduce cancer risks. And it was a triangulation of evidence approach. They put together lab data, natural experiments, human epidemiology, everything, and studies of antioxidants, and that this was a very reasonable hypothesis. It's the only time in Nature I've ever seen the editor put in a caution at the bottom of the paper saying unwary readers shouldn't take the accompanying article as a sign that the consumption of large quantities of carrots would actually reduce cancer risk, which was sort of good news, because when the trials were then done, trials following people up, massive numbers of people who were supplemented with beta-carotene, if anything, they had a small adverse effect on cancer. And there was just so many of these things, and not just in the dietary field. It's like HDL cholesterol, which is so-called good cholesterol, which is supposed to be a cause of some protect against coronary heart disease, doesn't protect against coronary heart disease. All of these are things that observational epidemiology said would likely to be major contributors to coronary heart disease, cancer, etc. And when the trials were done, everything turned out to be a bust. None of them worked. I'll skip that. So Paul Brennan from IARC and I updated the estimates just for cancer, because the data are easily available. And if you do the estimates today, you get 80% is the same estimate for preventable in principle. Now, since 1981, the two most important novel identifications of causes of cancer were HPV and cervical cancer, and H. pylori and stomach cancer. So both obviously infections. So one would think, how could it be that the proportion that is preventable in practice hasn't increased? It should increase, because we've identified some new causes, and vaccination works for HPV. And the reason it's not increased is because smoking has declined. Smoking was a great way of being able to have an identifiable cause of cancer. And because people are smoking less in the UK, there's 75% of the cancers in never smokers. We don't know, we wouldn't know how to prevent them. So as I say, there's nothing particular about cancer. It's just you have the data that allows you to do to look at cancer. For some diseases, the degree of missingness might be exaggerated, particularly when you're putting in a grant application. All grant applications in coronary heart disease used to start with, we can only explain 30% of coronary heart disease through conventional risk factors. actually at the population level, it can explain vastly more than that. And I did confess to the fact that I'd used this in grants in the past when you wanted to get money, but and this is for coronary heart disease, it can explain a lot. For type 2 diabetes, these are the results of the GLP-1 agonist trials of the incidence of type 2 diabetes. Adiposity contributes massively to type 2 diabetes, but for many causes of disease, we just don't know. The problem is, if you read the Lancet, which is a foolish thing to do, you'll assume that we know the causes of everything, because the Lancet has a series of articles called Lancet Commissions. When they get they get famous people, usually sort of towards the end of their career. They never invite me, that's why I'm doing so. Near the end of their career to say things about, and you get these ridiculous things like with dementia, they say, we know we could prevent half the cases. It's just simply absolutely not true, especially for Alzheimer's disease. So a large proportion of the risk is clearly, as I say, modifiable, but we don't know what it is. So why might that be? Well, firstly, most of the studies, like the Nurses' Health Study, Health Professional Study, UK Biobank, etc., recruit the middle-aged. And, maybe it's too late for identifying causes. Because, for many conditions, in particular neurodevelopmental conditions, very obviously, many psychiatric conditions, etc., it's really obvious that the disease process is underway from very early on. And increasingly, there are diseases, stomach cancer, peptic ulcer, multiple sclerosis, etc., where there's good evidence that things are happening early. In cancer itself, there's been well, it's not going over the top to say it's a paradigm shift. I think this is one of the very few things one can use that term properly. It's a complete revolution in understanding of cancer in the last 5 to 7 years, which is finding that when you sequence normal tissue, you've got driver mutations are there at age 10, for example. So, and because cancer within humans is simply a Darwinian process occurring within one body, you can date back in the way do a coalescence. You can date back to when the initial mutation in any particular cancer occurs, and in renal cancer, the mutations seem to be at age 10. Colorectal cancers sometimes even earlier. Same with endometrium. Basically, right the way across the tissues. So some things are going on early in diseases. For lots of diseases, evidence points to early life acquisition. So, for example, birth cohort effects are a very standard way of suggesting that. If you see whether the risk of disease depends upon the birth cohort you were born in, or depends upon the period through which you are alive. And sometimes some of these are very obvious, like they were for peptic ulcer. But with his way back in 1962, you actually see that for both gastric and duodenal ulcers, it was the birth cohort you were in, not the age you were at. That was that determined your risk, and this was going down earlier in duodenal ulcer than it was peptic ulcer, and it's now the stomach ulcer. So it's now thought to be H. pylori, and obviously treatment of H. pylori, eradication of H. pylori does indeed solve peptic ulcer problems. Stomach cancer declined, and no one knew why stomach cancer declined. Everyone speculated it was diet, but the disappearance of H. pylori is likely what drove it. I'll skip that. On that. For other conditions, life course Mendelian randomization suggests childhood is an important etiological period. Breast cancer is a particular one where larger prepubertal breast cancer, larger prepubertal body size is protective of later breast cancer. And you can separate early life body size, prepubertal body size from adult body size. And there's a really substantial effect of larger prepubertal body size lowers later breast cancer risk. coronary heart disease and type 2 diabetes are the exception, but they're the things that most epidemiologists now on Lancet commissions grew up with. That's what epidemiology was. It was cardiometabolic disorders, and they are perhaps exceptions, but they obviously drove a lot of the field. And also, there's many RCTs show that adulthood interventions don't work. So one reason you might not be able to identify these causes aren't being identified, is that within contiguous geographical regions, within populations, they're essentially ubiquitous. I.e., if you're doing an individual level study like UK Biobank, you're not actually getting enough variation in exposure, even if you could measure the exposure. An example of a ubiquitous exposure or near ubiquitous is cigarette smoking. In Dolan Hill's 1950 paper, probably the most remarkable number, I think, is that these are the controls. These aren't the people with lung cancer. Amongst the controls, 4% of the male controls were never smokers. Everyone had smoked. You got free fags given out after the First World War. All males, essentially, of those generations were at least smokers for a time. And Geoffrey Rose elaborated, as he often did. This is, if you read one paper, if you've not read this, this is the nearest epidemiology to a theory. It's only 7 pages long, beautifully written, about this notion. And he suggested this sort of thought experiment. He didn't have Bristol involved in the thought experiment, but some Bristol involved. You imagine that everyone smokes 20 cigarettes a day. So, a dictator takes over in Bristol and you've got to smoke 20 cigarettes before you're allowed to go to bed. You get your packet of Capstan full strength, you smoke your way through it. You can't go to sleep until you've smoked exactly 20 cigarettes. And, and then in Bath, no one smokes at all. Bath is very posh. Probably most people spit smoke anyway, but the dictator stops anyone smoking a single cigarette. So smoking in Bristol, every person smokes exactly 20 cigarettes a day. Bath, no one smokes at all. Then you follow up Bristol and Bath for 50 years, and Bristol would have about 15 times the lung cancer rate that Bath has. But within Bristol, smoking can't relate to lung cancer. Everyone's smoking exactly 20 cigarettes a day. So a study in Bristol wouldn't show any difference in cigarette smoking between the lung cancer cases and controls. And same within Bath, a few people will get lung cancer. No one smokes at all, so you can't be identified. But smoking causes essentially 90% of the cancers, of the lung cancers at least, probably more. So the population risk, what would come up as preventable in principle, be explained by something that if the exposure is truly ubiquitous, you just simply can't pick that up. And between the individuals, like in Bristol, the individuals who get lung cancer and not, the factors, people say genetics, but lung cancer is a bit heritable, but most of that is through, a chunk of it's through heritability of smoking. Chance is probably the most likely thing. Here we've got Winnie. This was from The Sun. I found this on a train in 2007 and tore it out. There was the days when people used to read newspapers and one would want to find out what population was thinking by reading the Sun, by picking up the unread copies of The Sun. And there he was. Here's Winnie. She smoked for 93 years and she ain't quitting now. So she's lighting a cigarette off the candle on her 100th birthday cake, and she's smoking away, and they're great, So the tobacco companies would say, let's sequence Winnie, and we'll find out the gene which makes you resistant to lung cancer, or whatever. Look at other factors in Winnie's life, but these are likely just to be essentially stochastic, just chance. So, when she was 60, mate, say, the postman is coming to deliver her birthday cards, knocks on her door. At exactly 11 a.m, she opens the door, gust of wind comes in, she coughs and she coughs up a metaplastic lung cell, she just dislodges it. And because of that, she's alive at 100. If the postman had come at 1 minute past 11 or 1 minute to 11, there'd be no Winnie. Winnie would not be there lighting lighting her cigarette from that cake. But the one thing we do know is there would be statistically there would be another Winnie who was lucky when she was unlucky. So the factors that you could probably never discipline, biological and social behavioural, just events, stochastic events, probably explain the difference. And then possibly for epidemiologists, especially ones who aren't reaching retirement, probably the most disturbing thing about thinking about how epidemiology can find causes is thinking about bilateral cancers of bilateral organs. So consider kidneys. You've got two kidneys. Now those kidneys share identical germline DNA, what they were given at what was achieved at conception. If you eat healthy fruit and veg, both kidneys will get the antioxidants distributed to them. If you smoke, the nasty stuff will get to both kidneys. If you drink, the nasty or nice stuff will get to your two kidneys. If you jog, the same thing. If you're unlucky enough to be given the anaesthetic Trilyn, which was, when I was a medical student, was used as an anaesthetic for pregnant women who were delivering. It's a registered renal carcinogen. If you're exposed to that, both kidneys would receive the exposure. So if one kidney develops cancer, you have this perfect control that has had every single exposure, virtually, you can think of. It's had every single exposure. And despite having had every single exposure, the risk in your other kidney developing a cancer is only about 50% higher than any random kidney in that population developing cancer. That is when it's matched on every exposure that we know and we don't know, that differs within the population. Now that really is sort of, let's just give up the job when we think about that, about trying to search for causes in that situation. But we do know that kidney cancer is something which shows huge differences between populations. This is cancer of the body of the kidney, not pelvis. I mean, there are a few things, trauma, therapeutic radiation, cosmic rays or whatever, that might have affect one side and not the other. But we can rule those out, I'll show later. And now there's nothing special about these being bilateral organs. It's just that you have the perfect control of the bilateral organ. The same will be the case for lung cancer. But you can't separate, it's very difficult to separate out whether it's actually a metastasis or it's truly a new primary. Same for colon, regions of the colon, etc. This will be a general phenomenon, but it's just the bilateral organs gives you this beautiful matched control situation. And and Richard Dole indeed pointed this out, saying, he was really, it was very surprising that women who develop cancer in one breast have a higher risk of developing cancer in the other than women who were previously unaffected, but the excess risk is small. Now, there's many vector issues, which I won't go through, about looking at second primary tumours in the other organ, including obviously, one is how many organs are at risk. Some very early epidemiology made a big mistake because, of course, a lot of those studies would only take the second primary as being in the other organ because it was difficult to separate out local spread from it being a second primary. And of course, if you've got cancer on one side, you've only got one organ at risk. So you've no longer got two organs. So you've actually got to do an obvious division to take into account the risk you see. And when one cancer is found, then surprisingly large, you find a sort of cancer's already existing in the other organ at the same time, you count that as a new secondary. You don't, you get increased surveillance. If you've got cancer, either metastases or true second primaries, etc. Treatment, such as preventive therapies, including prophylactic mastectomy, will obviously have a big effect. But taking those all into account, however extreme you make your estimates about their effect, this doesn't change the basic picture that the risk is relatively small. And most of the factors that we think of as causing breast cancer are things that are going to be true, are going to have the same influence on both breasts. So breast cancer is preventable in principle. There's large differences between populations. But one thing you see is that there's a near constant risk in non-affected monozygotic twin from the age her affected twin developed breast cancer. This is another one of those absolutely remarkable findings. Now, if you diagnose very early on a breast cancer, then your relative risk for having a second tumour is pretty high. But there you have the BRCA1, you have the people with monogenic disease conditions, etc. They're a tiny proportion of all cancers. So you do have quite a big difference in the general population incidence. But once you get to 60 or 70, there's really pretty trivial differences. The elevated risk, I say, across all ages, it's 2. The risk of 2 for cancer, for breast cancer. And this is the incidence in the patient's other breast. And of course, the monozygotic twin, who has 2 breasts at risk, has double the risk. So if you're a monozygotic twin and you've had a breast cancer, you've had a breast cancer, you are less likely to get a second primary breast cancer, even though you're perfectly matched on all those exposures. Than is your monozygotic twin. The near constant risk is, I'll skip this. This is of interest to cancer people. The fact you have a constant risk suggests that there's just one factor. There's just one thing that needs to happen. So when one thing needs to happen, it's no longer. It's just you tossing the same coin all the time. So you just have a constant risk of it happening. So it's to get this absolute risk. So, as I say, you need to consider it in the age distribution. So most of the cancers that are occurring in the population are occurring at an age group, in the age groups when there isn't this very high risk from BRCA1 or some familial cancer situation. And we know that they are, in principle, they are preventable. This is fascinating, but I'll skip it. Okay. There's now one study very recently done where when people had just lumpectomy, you looked at they actually measured the volume of the remaining breast tissue, and then taking the volume of remaining tissue at risk into account, calculated the risk of a second primary occurring in the other breast or occurring in the same breast, and they were the same. So there isn't some sort of field cancerization process which occurs. It's something happening in one breast. This cosmic rays couldn't produce that. Cosmic rays would you'd expect the cancer risk to be greater, or trauma, or whatever. Any of those one-sided things you'd expect them to be greater. So this is the important thing: is you think of pairs of kidneys in 2 populations within which exposure levels are ubiquitous. The only explanation which makes any sense of these data is that for kidney cancer, there's a 5-fold difference between reasonably large-sized populations. We've got good data from cancer registries, so we know that the 80% is preventable in principle. So you have the population where there's 5-fold the risk, and the population where there's the lowest level of risk. And if you're this person and you develop cancer of one kidney, basically your other kidney doesn't have much greater risk than the kidneys from the population because everyone's exposed to the same level. That's why you can get these. That's why you can get this really rather extraordinary findings. The only explanation that I can think of that makes any sense at all is, and I'll be interested in any other explanations for these extraordinary data, is that you have the differences is between populations. So now I'm getting to infection. I'm going to infection now for 20 minutes. Then could have been 30 minutes. But I haven't fucked up with the timing. So because one possibility that has been advanced with breast cancer is that the reason that your other breast doesn't have any sustained increased risk is that it's a short-acting, so-called the hit-and-run hypothesis, and this has been suggested for infections. People have suggested EBV infections. For example, you get EBV infection, and then for a very short time you increase the risk dramatically. The risk of developing cancer is increased dramatically, but then but that risk rapidly disappears. So I find this controversial. I'm glad I didn't read about this stuff when I was starting out in epidemiology, because maybe it would have been a good thing. I would have done something useful, become a tax accountant or something. So ubiquitous exposures, those that are similar for people within geographically to contiguous regions, and thus the variance between individuals within regions is low. Measurement error is a major problem. Chance is the major driver of things and risk within populations. So infectious agents are an example of ubiquitous exposures, particularly if you take the sort of infectious agents which everyone gets, but which the effect on disease is modified by age. You'll then get you'll get some distinction, but it's incredibly difficult to measure because the age when one first encounters an infection will vary, but there's no real sensible measures. There's very few data which allow you to look at that and the outcomes of that of when people actually acquire infection. But there are other ubiquitous exposures, but the age of infection is the one that I got most interested in. Thought had most mileage, which is why I'll move on to that. But there are other ubiquitous exposures are possible. So so thinking of age of infection, there's some real standout sort of historical examples of the huge modulating effect of age of infection. Yellow fever is one. Yellow fever is then totally endemic. Had been completely endemic in many populations. Therefore, as you do with endemic infections, it's usually young children who get the infection. It does increase your mortality risk, but it's a relatively small increase in mortality risk in young children, and they then become immune for life after surviving yellow fever, which the vast majority will do. The people who came in aged about 22, who had not been infected, were of course the colonizing soldiers in Cuba, in the case of the US, in places in Africa and elsewhere, in the case of Britain and France and Germany and others. That's why yellow fever. The reason yellow fever was the infection that was looked at, studied in such detail, was it was killing with 20-30% mortality rates. It was killing the colonizing soldiers. But to go away from that history, but the effect is incredibly large. The differential response depending upon when you got infected with yellow fever. Polio, of course, everyone knows because of polio, and the infection was delayed, especially in the relatively wealthy in the 1940s, 50s in the US. It's a gastrointestinal fecal orally transmitted disorder which gives you a bit of diarrhea, etc. As a kid, if you get it, if it was a young kid, if you get it later, then obviously the risk of paralytic polio is greatly expanded. Well, no, I won't go into my next law, but this has been sort of well known. These things have been well known for a long, long time. Good papers on the age of infection, and probably the pin-up case, which has been by far the most studied infection, where there were clear age of infection influences, is Epstein-Barr virus, which even in wealthy countries, virtually 100% of people will acquire it by sort of 50 or 60, sort of 90% will. And the disease risks very strongly depend on age of infection and potential cofactors. So in rural Uganda, for example, virtually all children at the age of 2 have evidence of EBV infection, which you can measure with, of course, you have but with antibodies. And in wealthy countries, there have been a few cases where one's known that a young child is seroconverted, and very often they'll have no symptoms, or if they have any symptoms, they're real mild, flu-y type, coldy type symptoms. In Africa, with very early infection, if there's then a later if there's a cofactor existing in the environment, which in this case is well known, it's falciparum malaria. If malaria is holoendemic, then you have then EBV together with endemic malaria related result in Burkitt's lymphoma, the commonest childhood cancer in Africa. EBV is likely to be related to other lymphomas, although it's not so well, especially older age lymphomas, but it's not so well characterized. It's also related to nasopharyngeal carcinoma, particularly in East Asian populations and a few African populations. In wealthy countries, in the UK, EBV is related to infectious mononucleosis, or glandular fever as it's called. If you get it at 16, 17 when you go to university, and you suddenly encounter saliva to a volume you hadn't encountered before, then and acquire EBV at that age, then the result is a post-viral fatigue syndrome. So infectious mononucleosis is a post-viral fatigue syndrome, which goes with you often have palpable glands. Long COVID is a post-viral fatigue syndrome where they have a sort of general central typology, but with things that might be specific to the bug, and with long COVID there is taste and smell differences that persist, etc. But then you get infectious mononucleosis. But then, and this is one of the most remarkable pieces of epidemiology in recent years, if you get to be like 2022 and you're a US soldier and you've had blood samples taken way back to the 1950s and 1960s have been stored, and you have not got evidence of having been EBV infected, and then you do get infected, your relative risk of multiple sclerosis is 30. It's sort of the sort of things we don't see in epidemiology. So that's one. Now this EBV has been studied greatly. There are EBV is herpes virus, so those viruses are forever viruses. They reside, some of them in beta cells. It depends what the herpes virus is. But so these are the sorts of agents that one would think might relate to disease. So these are things you would want to study. And the characterization of age of infection of these is really poor. Even with EBV, I was amazed when I searched very hard to get the studies on EBV. The number of studies that actually can date at what age people first acquire EBV within countries, even that's by far the most data for that, is still relatively poor. Something like Kaposi's sarcoma herpes virus, where in Uganda about 40% of people will have that eventually. A lot of them get it as young kids, and you can show at a later age their antibody levels are much higher if they had very early infection. So this is a disease where it's thought that very early infection increases risk of all these things. So this is just trying to make the case that these are things that are worth investigating. I mean, hepatitis B is a sort of pin-up of the age of infection because you're more likely to get persistent actual viral infection. These don't have the forever quality of forever for everyone quality of most of the herpes viruses. HPV obviously persists. Then there's RNA viruses, which are generally not persistent with lifetime immunity, but which very clearly some of these have age of infection effects. Rubella has a very particular age of infection effect, which is you don't want to be exposed as a fetus because that's German measles and leads to the neurodevelopmental sequela of German measles during pregnancy. And then there's lots of other viruses. Viruses with waning immunity to waning at a particular speed. Coronaviruses were, of course, studied in the 1980s in the Common Cold Unit through inoculations. Actually, they did this, studies of actually giving people a coronavirus squirted up the nose. You could then show that the infection became established. The technology, obviously, for measuring antibodies was not sensitive. And after 1 year with the OC of the four seasonal coronaviruses, the OC variant in the Common Cold Unit, after a year, there wasn't a detectable antibody in anyone. But when they re-inoculated people, they didn't get ill. 4 of them actually got an established infection, but they didn't get ill. So there was obviously some immune memory, but that immune memory wanes further after that. So, and waning immunity might eventually lead to a state that's similar to initial immunological naivety. So we were saying about the fact that if you get infected with yellow fever when you're very young, then that has a different effect than if you're immunologically naive when you're 22. But that's for something where you get lifelong immunity. And this is the sort of pattern of the seasonal coronaviruses from the US data, when they weren't be collecting any more of that nonsense anymore, because obviously viruses don't matter. And it's the ultra-processed foods which we need to stop eating. That'll make us healthy. But so with these sorts of patterns, when you look at and you work out of things arriving, some people will go long periods of time just by chance and not get infected. Some 16-year-olds or 20-year-olds will actually get to that age without having, without having one of these. And then if they get infected at that later age, this might lead to fatigue syndromes that are seen. So chronic fatigue syndrome the whole thing might be a whole package of post-viral illnesses, but where the virus does modulate to an extent the symptomatology, etc., but it doesn't actually drive the central feature of it. So they could result from exposures to ubiquitous viruses for which age of initial infection is late, or immunity is waned for a long period. And there's evidence for that in the other end of the life course at the older ages. There are these sorts of reports. There's scattered reports over the pre-COVID period where care homes would suddenly be increased deaths or very serious illnesses in care homes, and the Epidemiological Intelligence Service or whatever goes in, and all they find is a coronavirus, rampant in the whole population. Probably that you had a place that hadn't been visited by this by chance by one of these bugs, and then when the bug arrives, the people are immunologically naive and do badly. Not just viruses, but I'll skip on to there's obviously other important infections. So what do we need to know? As I say, the extent to which age of initial infection and stroke infection when immunologically naive are important influences on population health needs to be known. Now, this could be studied at the other end of the life course with waning immunity, but I think it's probably more. It's going to get better evidence, or there's going to be more going on at the early ages when first encountering these infections. So we need to have normative data for age of initial infection, then trajectories of immunological response, including waning, health-related outcomes when immunologically naive, and the extent to which this contributes to the missing environment. This could be a major contributor. Things together, this could be a major contributor to clearly preventable and invincible diseases. So as I say, these things are things that differ so greatly between populations. In rural Uganda, you won't find anyone at 8 over 3 who hasn't had EBV. In the rich countries, these are getting this portion who get to older ages without the infection grows and grows. So getting data on that is a very, very considerable challenge, and there aren't any studies that exist which actually can provide data on that, because Alspac's the best birth cohort in the world. Everyone knows that, but for Alspac, we first blood sample we got across the whole cohort was at age 7. Tiny subsamples had earlier sampling, and then we only got it 2 or 3 more times before adulthood on the whole cohort. So you can't just look at existing cohorts. But there's the opportunities in the UK are first the adolescent health study. So the adolescent health study is getting a very substantial amount of cash, at least 60 million just to start itself up. And the adolescent health study is predicated on the obsession of Keir Starmer and other right wingers of people trying to avoid work. Kids, you have all these kids who are not going into work, these adolescents that don't contribute to the growth of our great nation, and that's a real problem. So we've got to do something about that. And the reasons for that often seem to be also mental health issues, fatigue issues, etc. are issues which play into that. So the adolescent health study has been set up as a study of mental health, but essentially. It's called, they're also going to say that mental health and being overweight, those are the two issues. But they're going to sample 100,000 kids. They're going to take them from school years from 8, 9, 10, 11, 12, 13 up to 16 or 18. They haven't decided on the final age. So that's quite a lot. That's sort of 10,000 in each year group, and then follow them up every year. Now they haven't got any money for blood sampling, but consider it compared to actually setting up a study de novo just to look at age of infection. This is, of course, brilliant because you're going to get follow-up on all of these kids forever. They're not going to stop it. Once a cohort gets set up, until the people who are attached to it die, they carry on usually. Kathy Sudlow is the CEO of that, and is very interested. We had a meeting with Kathy and 15 other people that work on infection. She doesn't work on infection. Who work on infection to try and get over that this will be an important thing, and she definitely thinks it will be an important thing. And then that only gives us from age 8 up. So we need to get 0 to 7-year-olds, and they could come from a selection of the birth cohorts that are recruiting. Alspach is getting the children in the 90s, a few hundred a year. It's not enough. But there's also the Bradford cohorts, which are also recruiting for new studies. There's Seagull, Louise Kenny's cohort, and others' cohort in Liverpool. So we could easily get, across the birth cohorts, we could easily get 1,500 in each age group, and we could get 10,000 in the adolescent study. Now, it's expensive doing assays, but you could at least do a subset. You could take samples, including dried blood spots from everybody, and you could get venous samples from, say, 1,000 each year, or enough to give you 1,000 at the end of the follow-up. And now that's expensive, but it's not. But compared to actually setting up a de novo study, I think setting up a de novo study is impossible. So until when I saw the adolescent health study being announced about 9 months ago, I rang back Kathy, 6 months ago. No, it was probably 9 months ago. So this is something that could really be added to it. So you've got to think what data and biosamples are already available. You need to get data from geographically disparate settings. So we're going to Uganda. So the idea is we set up a companion study in Uganda, and Kenya are interested. People in Kenya are interested. So it would be good to get. You'd want to get data from more than one place, obviously, for looking at the geographical differences. Intrauterine infection is important, and need to think about how that will be dealt with in the birth cohorts. That's John's flagging me off as he walks out. At least so. What I'm just saying,
I haven't got time to ask my question because you won't.
Okay. Assays. There are now multiplex assays available, of which probably those of you around on the 9th of July, Tim Waterbauer from Heidelberg, who's the person who set up the assay array for UK Biobank, will be here in the talk. And there's uncharged assays. I'll skip that. Family recruitment will be great because infections come from other people. So it would be possible to get at least in the intensively sampled 1,000, it'd be possible to get that family. And you could archive samples on larger numbers for future case control analysis. So if you get dried blood spots, and then you can go back, and when people get fatigue, you can go back and get their set of samples over time, coming up to that. And then what would be particularly powerful, but there's no insurance that it will actually work, but there's a lot of interest in the notion that epigenetic measures might produce long-term measures that might actually relate to the age of infection in the way that they relate to mother smoking during pregnancy, for example. Certain infections, I think, very exciting because you can't date it, and in current studies you just can't date when infected. And if you look at rodents, if you inject a bolus of IL-6 interleukin-6, which is a big shock, but if you inject that at different days during pregnancy, for the first few days, no effect. You give it for a short period, and you get just completely scrambled rodent brain. And then you give it after that period, no effect. So, infections stimulate IL-6, so that's why IL-6 has been the notion for particular psychiatric conditions, for example. So large spectrum epidemiological studies aren't that great. In these international studies, we need to go back to ecological sampling. These are mutation signatures that you can get from that tell you about exposures. And this is a remarkable situation. Japan has a ridiculously high rate of kidney cancer compared to any other East Asian population. They shouldn't be getting kidney cancer, and they've got very high rates of it. And this is a Paul Brennan study where they sequenced the tumours, and in Japan, 72% of the Japanese cancers have a very distinct mutational signature. Now, these mutational signatures are easy. They often have been mapped to things like tobacco, ultraviolet light, apostolic acid, aflatoxin, etc. So it's very likely that there is something in Japan which is ubiquitous, which is leading to particularly high rates in Japan, and nowhere else has this signature. So it's a remarkable thing. These are likely to relatively sub 30 for MS and EBV. I mean, you don't see these in epidemiology. Tissue-specific methylation data might be valuable. I'm just trying to support your career for the next 20 years, Matt. I'm going to get you through to 66.
Yeah, exactly.
67. So, I think that might be work. We need to work with environmental scientists. Ecology of animal populations are interested in. Mendelian randomization works. If you have something which is absolutely truly ubiquitous, so you have Bristol, 20 cigarettes a day. There's a genetic variant, which is the most strong genetic variant that relates to lung cancer. It's the one that comes out top in the lung cancer GWAS, and it comes out top because it means you smoke more heavily. If you start smoking, you smoke more heavily, and you inhale the cigarettes more, and you end up with higher cotinine levels, etc., and you get more lung cancer. So this variant would relate to lung cancer within Bristol, even when everyone's smoking exactly 20 cigarettes a day, and the variant would tell you, but it's the smoking that's doing it. Amazing. Great, this Mendelian randomization. And it works in Burkitt's lymphoma as well. That shows what a crap photographer I am, because that's my reflection holding up my pathetic iPhone. That's Burkitt's original map of Burkitt's lymphoma, showing that it was overlapping with falciparum malaria. So how can you do Mendelian randomization of malaria? What protects? What genetic variant protected? Sickle cell, yes. So there you go. You look at sickle cell, and it's protective. Decreased risk of Burkitt's lymphoma. It's preventive of malaria, falciparum malaria, but it relates to Burkitt's lymphoma. It's a beautiful story. Malcolm Pike thought of it first. So anyway, so age of infection might be an interesting thing to investigate epidemiologically in general because it would fulfil some of these criteria for sorts of exposures which appear to be causing disease that we can't find UK Biobank, etc. And then you can do things about it, like HPV vaccination, triangulation. These are the people who spoke to some of these people for more than 35 years on these issues. If you're going to do further reading, then this is the best introduction to thinking about epidemiology outside of Jerry Morris's uses of epidemiology. And I gave this talk, when I first gave a talk along these lines, I showed this, £1.53. I advertised it. At the end of the talk, I didn't know there was online options available, and my phone goes on Richard Peter saying, thanks for advertising the book. And then I went back recently and looked. It's now £132. This guy. So I need to make Richard a bit of money from this box full of unsold copies of Causes of Cancer. That's it. Okay. Sorry, I started late. I was next in an hour. Fully understand if people want to go, but I'm also very interested in.
So we're 5 past, so if you have other meetings, you can go, but otherwise, please stay, like, ask some questions.
Disbelief. I'd like some disbelief. That'd be good.
So you plan to test all sorts of that?
Yeah. So the idea would be to use the Heidelberg, this sort of asset, this multiplex assay system they have. I mean, that the issue about these things, of course, the price is changing. The more you do, the cheaper it is. There's obvious candidates like the herpes viruses would be obvious. I'm interested in the seasonal coronaviruses, and because of COVID, they've set up you can now get a seasonal coronavirus. You know, they used to be very difficult to get and expensive to get seasonal coronavirus serology done well. The issue, it depends on the bug, how many. It's in plates of 48. So you have to work it out. So something like H. pylori, you need sort of 6 antigens really. But you can. There's the alternatives. I think of VeerScan, which claims to do 1,000, which sounds fantastic of a small spot of blood, but there's never been any validation data published on it. Although it was used as the initial scan in the MS study. They did run VeerScan and EBV did come up, so it's obviously not random.
Have you seen there's also a HIV link with MS as well?
Oh yes, and yeah. I think one of the Goldacre family.
Yeah. It was in his picture. So.
Oh right. Yeah, no, I hadn't seen that. Yeah.
I know it's a health study. Do you know whether they kind of do omics measures as well?
Are they going to do
Omics?
They're not. They weren't going to. They're basically a heartbeat setup. It's to ask people how they're feeling. So
I'm just asking. Yeah. So it would be really interesting to.
No, if one can get blood, then obviously the dried blood spots are more realistic on very large numbers because of the storage cost is just huge. It would just be crazy to store if they didn't store samples. We obviously want DNA. That's easy to get. But they're going to. So the infection story is the one that they're going to pilot because they're interested. As long as we got the 8-year-old.
And in terms of outcomes,
No, sorry. As I say, you might have mentioned. I think skipped on to the 90s stuff. The recruitment strategy for the adolescent health study intended to be through
Schools, yeah.
And selected schools based on. Is it? It's sort of you did at the end, so like the healthier and wealthier populations tend to take part. If they're not concerned, that'll be the same.
I think what they're going to do is oversample schools in poorer places.
Yeah.
Which so it's good. So that just means you have a captive audience. But if you're looking at the school refusal is thumping up. So if you're looking to be studying things like being not in employment, whatever NEET stands for.
As you know, we infectious disease modelers spend all of our time fitting in infectious disease models to data to estimate the course of infection, which is related to the inverse of the average age of first infection. So I wondered whether you could do some modelling to predict what the distribution or what the average age of infection is going to be for all the things that you're looking at.
Yeah,
who looks.
I mean, no, but that's why I sort of wanted to talk to people who know about what sort of. I know we had a brief chat about cluster, for example, about what, because this surely would be fantastic data for modelling.
Yeah, but I mean, some
and-
pre-modelling, but because
Yeah.
it would be interesting to know how good the predictions for the average age.
Yeah.
Just for course.
Yeah, exactly. Do it in advance. Yeah, that would be very
And that's
interesting.
well, but maybe you could target, maybe target something where you should be sampling for different, or what age group you should be sampling for different.
Yeah. No. I mean that's absolutely right. I mean, yeah. I mean, that's a really good idea. I mean, like in terms of, it's not worth measuring something at an age when no one's got it yet, for example.
Yeah.
So one would want to think. So the advantage of this sort of the sort of Heidelberg thing that allows you to do sort of bespoke things at different ages. That's a very good idea. But has anyone sort of? I just couldn't find any sort of. I'd be very if there's any sort of reviewy papers or papers that sort of show that. I found some sort of malaria thing from.
There's literature on age of
Yeah.
Actually, we have a PhD student who is related a bit to the waning stuff that you're looking at. He's developing within host models, and then like naturally it comes out of that the time between infection, which would be related to what you were saying.
Yeah, about waning. Yeah.
Waning and length of time between infections.
Yeah.
I'll send you a picture.
That'd be great. One of the papers I put up, there's this Paul Master. This it was an extraordinary amount of work with Judith Glenn. There was I talked too much. That we go. I don't know if you've seen that one. The systematic analysis of infectious disease outcomes by age, which they, but of course, virtually all of the studies they are reviewing are through notified, infections that are clinically diagnosed or notified or whatever. There's very few. There were very few studies that actually had any sort of immune measure, antibody measures, for example.
Yeah. Thanks, George. It was very interesting. I was just wondering, if I understood it correctly, you mentioned coronary heart disease is one of the diseases where the risk factors, they mainly affect you in adulthood. But doesn't a lot of risk factors probably affect you before, if you look like at MR, LDL cholesterol, for example?
Absolutely. I mean, when the Korean War, the US soldiers at 19, when they were autopsied, which was at the height of the US CHP epidemic, had their own at 19. The disease is developing before, but you can reverse it later. I mean, that's as you know, sort of statins. You get about 35% of the lifetime. 5 years of statins achieve about 35% of the difference you get through a lifetime lowering cholesterol, for example.
Yeah. Okay, so that's the important difference.
All right, I had another question about what impact COVID would have on it if you've got a break where people didn't get infected.
Yeah, but I was giving a talk in Copenhagen, and there was someone. Denmark has very good sort of registries, but they're not. But again, they all require someone notifying them of the infection. They were interested in saying that, looking back to see how things changed. I mean, it was a sort of natural-ish sort of experiment, but the problem is it was a natural experiment which influenced many things, from school refusal to infection and all those others. But it definitely would give it should give sort of perturbations that could be followed up. Yeah, definitely.
In terms of the adolescent study,
Yeah.
given that Kathy Sudlow's kind of in charge of it? Is there any, you know, if there's anything to that link with the educational records?
Oh yeah, they're absolutely, they're definitely wanting to do that.
They've
So,
got a self-report, how are you feeling stuff, and
and then yeah, exactly. So the outcome, yeah. So education will, A levels will be one of the main, will be sort of particular outcomes that they'll be interested in, and so all the stuff that Andy Boyd, because Andy Boyd's been working on getting sort of linkage data for cohorts in general, so it'll be tied into that network. Yeah. Yeah, thanks very much. Sorry again.
Online, so
Oh, no, they're on.
I checked.
Okay.
Thanks very much.
No, thanks. And yeah, if people are interested in this, then that's nice. And certainly the modelling side, obviously, would be something. I mean, there's obviously a lot to do with the data, but this will be done in the usual way of the data being available to everyone. It gets funded. Whether it will get funded, be interesting.
So what's your plan on that?
I'm talking to Aria. Aria are interested, so not through grant, no, but the gates. Trying to find people to scrub up and send them to talk to. Gates. And it's not my natural skill. But I think it's going to have to be funded that way. But we do think that we're trying to welcome like for money to be piloting it, etc. And the main thing is just get enough Gosh to get at least get some start get blood spots once it starts being seen, because once they've gone, they've gone. And it's not gonna happen. So the reason I about Uganda is I was thinking the only place where it would be plausible to do a study would have a health study was Uganda, because sort of population surveillance. HIV, of course, they've collected samples, but they've got freezers and freezers full of samples going back a long time. They did the closest psycho and herpes virus stuff, different ages of kids. Yeah, but I don't know how many of them kept samples. That's the thing. I think Uganda Virus Research Institute, it's not particularly, but Caliphi is meant to be just like to check it out. Yeah, those sort of places. So
Split spots, or is it? Do you need?
yeah, no, you can use blood spots. Obviously, you can do more plates. The more sample you get, then obviously prefer venous samples. But during I was part of the ONS study, and we were trained to prick ourselves and get enough blood into tubes, and it's not that. So self-collection of samples would be a possibility, but as always, the problem is that the people that you want to respond are going to be those most likely to not respond.